Hi everyone, this is Ratna. Welcome to Basics of Biotechnology. In this tutorial, I am going to discuss about PCR. PCR. It stands for polymerase chain reaction. It is a revolutionary method developed by Carey Mullis in 1980s. It is a method widely used to rapidly make millions to billions of copies of specific DNA sample. It allows scientists to take a very small sample of DNA and amplify it to a large enough amount of study in detail. See, this is a double-strand DNA. It involves three steps: denaturing stage, annealing stage, and extending stage. In denaturing stage, the hydrogen bonds between the two strands will break and the double strand will separate. This after denaturing stage, it will go to annealing stage. The primers will attach, forward and reverse primer will attach at the end of these two strands, and after attaching the primers, extension will take place at the temperature of 72 degrees. This method can make millions of copies of DNA. It totally depends on base pairing of DNA, which is A with T and G with C. See, PCR is similar to the DNA replication only. Both are making millions of copies from the target DNA. You need to know a bit of sequence at the start and at the end of the region of DNA if you want to make a copy. You make primers which will match the sequence. This is the only part of the reaction that is specific to the target sequence. Primers determine the size and sequence of the PCR product. You also need Taq polymerase, which polymerizes the reaction, and PCR buffer, and magnesium, which is tag enzyme needs this magnesium content. Magnesium, if we increase the magnesium concentration, it will affect the specificity of the PCR. DNTPs, deoxynucleotides A, G, C, and T to go into the PCR product, template DNA. The sequence you are going to amplify the product from. This is the target DNA. There are three steps involved in PCR: denaturation, annealing, and extension. As I already told you, if we apply heat to double-stranded DNA, it is easy to break the hydrogen bonds and to make it single-stranded. The same thing is happening in denaturation step. We are applying 96 degrees of heat to this DNA double strand to make it single-stranded for the next step. In annealing, we are cooling the reaction so the primers can bind to their complementary sequence on the single standard DNA template. See, after separating the two strands, the primers will bind to this complementary sequence of on the single standard DNA template. After primers will bind to the single strands, what happens is extension will start. It will raise the temperature so that the tag polymerase extends these primers, synthesize new DNA strands. In these three methods, annealing temperature is the most important temperature. We can't change the denaturation and extension temperature, but we can change the annealing temperature. It totally depends on the primer which binds to the template sequence. Reaction is carried out in a PCR thermocycler. Usually it is 35 cycles. It is an exponential, very quickly get larger copy numbers of the PCR product. In denaturation, hydrogen bonds are breaking, and at annealing, primers are attached, and in extension, Taq polymerase extending these primers. See, this is the template DNA. For each cycle, the copies are increasing rapidly. See, in first cycle, there are 4 copies, and second cycle, it is 8 and 16 and 32 copies. In every cycle, the copies of the template DNA is increasing rapidly. After completing the PCR, we looked at the products with gel electrophoresis using agarose or acrylamide gel. Gel is a porous polymer which impedes movement of DNA based on the size. See, look at this gel. The DNA is at different sizes. It moves based on the size only. See, this sample is around 400 base pair and it is around 100 base pair. There are various sizes of DNA samples. And DNA is negatively charged, so we apply an electrical current and DNA will run towards a positive electrode. We can identify gene deletions, insertions based on these PCR product sites. But however, majority of the mutations cannot be seen by PCR, so we need to do sequence of the DNA.